Our class at Calvin University is excited to announce the isolation of 7 bacteriophages on the host bacteria Gordonia terrae 3612 in fall of 2021. 4 of these phages were selected for genome sequencing: Gezellig, Phomeo, Excresco, and Barbochs. We have completed our analysis of the genomes of Gezellig and Phomeo, and we plan to look at Barbochs later this semester. Excresco, however, was found to be a duplicate of a previously discovered phage. Our research this semester has therefore covered 3 topics: Gezellig's genome, Phomeo's genome, and the frequency of the discovery of duplicate phages. Gezellig, the first genome we looked at, is a DC1 cluster phage. Fascinatingly, this phage differs from the Phages Wizard found in Pittsburgh in 2015 by only 20 nucleotides. Of these, 16 are located in ORF78, which at this time has no known function. Of these differences, 7 resulted in changes in the amino acid sequence of Gezellig compared to Wizard. These can be seen in the image in the bottom left, with the yellow areas representing identical nucleotides and white showing differences between the genomes. The sequence of ORF78, along with the sequence of the equivalent ORF and 21 other phages in the cluster, were analyzed with Selecton. The 2 other phages in the cluster were not closely related and did not merit inclusion in the comparison. All amino acids were found to be undergoing either negative or neutral selection. The bottom left corner shows each amino acid in ORF78. Amino acids in pink are undergoing negative selection, with darker shades indicating stronger negative selection. Amino acids that are not highlighted are undergoing neutral selection. As you can see, no amino acids are marked with yellow, which would have indicated a positive selection. In addition, we analyzed the genome of Phomeo, a CR2 cluster phage with 94 ORFs. It was also nearly identical to a previously discovered phage, GIFL16, found in 2016. The only major difference between the 2 was Phomeo's ORF20, a minor tail protein, which is swapped out for a different minor tail protein in GIFL16. No specific evolutionary pressures could be determined that would account for this change. We discovered that within the CR2 cluster alone, there are 5 different variations of this tail protein. The image on the top right of the poster shows 5 CR2 cluster phages, each representing a different variation of this ORF. We concluded that within this cluster, the minor tail protein appears to be a region subject to high rates of gene swapping. Finally, we investigated how common it is for duplicate phages to be discovered. Our phage Excresco was found to be a duplicate of Gambino, a CV cluster phage found in 2017 in Pittsburgh. This is a somewhat unique case within Gordonia terrae 3612 phages, as no other duplicates have been found across state lines. Additionally, another duplicate of Gambino, MattSquared, was discovered in 2021 in Pittsburgh. Because of this, we decided to conduct an investigation into the frequency of the discovery of identical phages. We expected that as the number of phages sequenced increased, the likelihood of a newly isolated phage being a duplicate would also increase. However, upon analyzing the data in Phages DB, we discovered that this was not the case, as shown in the graph in the bottom of the middle section. We also found that bacterial hosts in the Gordonia genus have more duplicates than all other hosts, with almost 4% of sequenced Gordonia phages being duplicates compared to 0.65% of phages isolated on all other bacteria. The duplicates found using Gordonia terrae were exclusively found within the 3612 strain. Because of this, the percentage of duplicate phages isolated on all strains of Gordonia terrae was smaller than the percent on strain 3612 alone. These results are shown in the graph in the middle of the poster. After looking at the percent of duplicates in Mycobacterium, Streptomyces, and Mycobacterium hosts, we found that the hosts used most frequently tend to have more duplicate phages. In sum, our research has determined 3 major takeaways. Firstly, Gezellig is quite similar to Wizard, and of the nucleotide differences between them, 80% occur within a single ORF. However, none of the individual amino acids produced by this ORF appear to be undergoing positive selection. Additionally, within the CR2 cluster, the minor tail protein appears to be a gene subject to frequent gene swapping compared to the rest of the genome. Finally, we determined that the Gordonia terrae host bacteria has a much higher percentage of duplicate phages than other host bacteria, and that the number of duplicates per year has not increased over time.